All right, get ready to dive deep because today it's all about integrins.
Integrins, yeah.
These proteins that are basically the glue and movers for your cells.
Mm-hmm.
Kind of like cellular parkour, just, microscopic style.
Yeah.
So we've got all this research piled up, ready to unpack it all.
Awesome.
How these tiny integrins actually get the job done.
Well, what's really cool is that integrins, we're talking way more than just sticky proteins.
Okay.
These things are like vital for a whole bunch of processes. I mean,
Yeah.
immune healing, immune responses,
Gotcha.
and even how an embryo develops.
Come on.
So just imagine, like, your cells are the bricks, right? Integrins are the mortar holding everything together to build, you.
Okay, I like that. That's a great visual. So speaking of teamwork, I see here integrins themselves are kind of a dynamic duo.
Yeah,
Something about alpha and beta subunits?
you got it.
Okay.
Can't have one without the other.
Mm-hmm.
These two subunits, the alpha and beta, they work together. They form this, like, bridge across the cell membrane. It's like a handshake, really,
Okay.
connecting the inside of the cell to the outside world, which is, the extracellular matrix.
Ah, so that's how they kind of anchor the cell. It's all about this handshake.
That's a big part of it.
Okay.
But it gets even more interesting, honestly.
Right.
There isn't just one type of integrin, see?
Oh.
Think of it like a toolbox full of different integrins.
Okay.
Each one with its own combo of alpha and beta subunits. And these different combinations give them, like, specific binding preferences.
Okay.
It's like each one is, specialized for a particular job in the body.
So, like, how do they actually, like, grab onto the extracellular matrix?
Yeah.
These sources keep mentioning this mysterious RGD motif.
Ah, yes, the famous RGD. Stands for arginine glycine aspartic acid. It's just a little tiny sequence of amino acids.
Right?
But it acts like a docking station for these integrins. Think of it as a special code, like, embedded in proteins within the extracellular matrix, and the integrin sees this code, and boom, it's like a handshake at the molecular level.
Got it.
Allows the cell to attach.
Okay, so RGD is the code. Got it. But do integrins just, hang out there all the time just waiting for a handshake?
Not quite.
Okay.
Integrins can actually change their shape.
Okay.
They switch between an inactive and an active state. Think of it like, a switchblade.
Mm-hmm.
When it's needed, it flips open and boom, becomes active and can bind to that extracellular matrix.
Mm-hmm.
And guess what triggers that change? Signals from within the cell itself.
Oh, so it's like the cell's telling the integrin when to reach out and grab on.
Yeah.
So there's some serious communication going on here.
Absolutely. And then once those integrins latch onto the extracellular matrix,
Yeah.
they don't just hang out solo. They cluster together
Okay.
and form these, strong anchoring points called focal adhesions.
Focal adhesions.
Yeah.
I'm seeing that pop up a lot.
Mm-hmm.
What are they exactly?
Well, think of them as the rivets or bolts holding, a structure together.
Okay.
They actually physically link the cell's internal skeleton, the cytoskeleton,
Yeah.
to that external scaffolding of the extracellular matrix.
Mm-hmm.
It's how your tissues can actually withstand mechanical forces
Wow.
and maintain their integrity. Without focal adhesions, everything would just fall apart.
That's pretty incredible. So integrins are like these tiny but mighty construction workers, basically.
Yeah,
Building and maintaining our bodies.
exactly.
But cells aren't just stationary, are they?
Right.
They need to move around, migrate to where they're needed. So how do integrins factor into that?
Well, that's another amazing thing about integrins.
Okay.
They're not just, static anchors.
Right.
They can dynamically regulate their grip, allowing cells to, crawl and migrate. Picture it like a slow, controlled climb with integrins acting as tiny hands and feet, gripping and releasing that extracellular matrix.
So like a carefully choreographed dance of grip and release.
Exactly.
Yeah.
And it's this ability to modulate their grip that allows cells to move where they're needed, whether it's for wound healing or immune response or even during embryonic development.
This is so intricate.
Yeah.
But I just want to make sure our listeners following along, why should they care about these tiny little integrins?
Think about it.
Yeah.
Without integrins, your cells wouldn't stick together properly.
Mm-hmm.
You wouldn't be able to heal from injuries.
Mm-hmm.
Your immune system wouldn't be working right.
Right.
And your tissues would literally fall apart.
Oh, wow. Okay.
Yeah.
That's pretty vivid.
Uh-huh.
So these tiny integrins are keeping us whole and functional.
Absolutely.
Wow.
And then it gets even more fascinating when you look at what happens when integrins go wrong?
Okay.
That's where we see links to all sorts of diseases.
Oh, really?
Yeah, from bleeding disorders to even cancer.
Oh, wow. So malfunctioning integrins can really have a domino effect.
Exactly.
Okay.
That's why understanding how integrins work, their structure, how they bind, how they're regulated, is so crucial for developing new therapies and treatments.
That's a deep dive thought for sure.
Yeah.
It seems like these integrins, despite their size,
Mm-hmm.
are holding the key to understanding some pretty big health challenges.
Absolutely. And that's why we're here, to delve into all that complexity.
Right. So to understand how things can go wrong, we need to understand how they work when everything's going right.
Yeah.
Let's zoom back in on those alpha and beta subunits.
Okay, great point.
Yeah.
We really need to understand how those subunits come together to create these amazing molecular machines.
Right.
What's been discovered is that these subunits aren't just randomly put together.
Okay.
They form very specific pairings. And each pairing, with its unique combination of amino acids, actually dictates which molecules in that extracellular matrix that particular integrin can bind to.
Oh, so it's not just a general handshake.
Right.
It's a very specific interaction.
It is.
Like a lock and key.
Exactly.
All right.
It's called ligand specificity.
Okay.
So, for example, some integrins are specialized to bind to collagen,
Yeah.
which is a major structural protein in our tissues. Others prefer fibronectin, which plays a role in cell adhesion and migration. So each integrin pairing has its own preferred dance partner, you could say, in that extracellular matrix.
I like that analogy. Thinking back to that switchblade analogy for integrin activation, what exactly triggers that switch to the open active state?
Well, remember we talked about signals from within the cell.
Yeah.
One of the key players here is a molecule called talin.
Talin, yeah. I think I remember reading about it. Doesn't it have something to do with linking integrins to the cytoskeleton?
Yes, but it's also super important for activating those integrins. Think of talin as a sort of intermediary. It can be activated by all sorts of signals inside the cell, like changes in calcium levels or the binding of certain signaling molecules.
Okay.
And once that talin is activated, it binds to the integrin's cytoplasmic tail, the part that extends inside the cell.
So it's like talin is flipping that switch.
Yeah.
Telling the integrin to extend and grab on.
Exactly.
Wow.
And this whole process is, tightly regulated to make sure that those integrins are only activated when and where they're needed.
That's incredibly precise. So it's not just about having integrins, it's about having the right integrins in the right place, at the right time, and in the right state of activation.
Absolutely.
Wow.
And that just shows, how complex and finely tuned these cellular processes really are.
This is all so fascinating.
Yeah.
But I'm realizing we haven't even mentioned that thyroid hormone receptors and integrins connection that's in our notes here.
Oh,
It seems like a total sidetrack from everything we've been talking about.
it might seem that way at first glance, but it really illustrates how seemingly unrelated systems in the body can actually impact each other.
Oh, okay.
Remember I mentioned that integrins are essential for cellular differentiation,
Yeah.
that process of cells becoming, specialized.
Right. Right.
Well, thyroid hormone receptors play a key role in regulating gene expression, which is, of course, crucial for that whole differentiation process.
Okay, I'm starting to see the connection.
Yeah.
So if there's a problem with the thyroid hormone receptors, it could mess with gene expression,
Yeah.
which then could disrupt cellular differentiation
Right.
and potentially impact integrin function.
Precisely. And that's exactly what research has shown.
Wow.
Certain mutations in thyroid hormone receptors, specifically the ones that lead to the loss of a crucial part called helix 12,
Okay.
they can actually disrupt cellular differentiation and potentially affect things like cell adhesion and migration.
Wow, that's incredible. It's like a domino effect,
Yeah.
One seemingly unrelated mutation having these ripple effects on other cellular processes.
Exactly. And that's why it's so important to study those seemingly isolated mutations and
Yeah.
understand their broader impact.
It really highlights how interconnected everything is
Yeah.
within ourselves.
Mm-hmm.
But before we go down that rabbit hole, let's loop back to those focal adhesions.
Okay. Yeah.
I'm still a bit fuzzy on how they actually form
Mm-hmm.
and their role in cell behavior. Can we dive into that a little deeper?
Absolutely. What's fascinating is that they're not just static structures. They're these dynamic hubs of activity,
Oh, okay.
constantly assembling and disassembling as the cell interacts with its environment.
So they're more like temporary campsites than permanent settlements?
Oh, that's a great way to put it.
Okay, I like that.
And the formation of these campsites, you could say, it's a multi-step process involving a whole bunch of proteins.
Right.
It's pretty complex.
Right, I'm ready.
Awesome. So first you've got those integrins clustering together, right?
Yeah, like calling in reinforcements to really grab on.
Right at the point where the cell attaches to the extracellular matrix. It's like they're joining forces to make a stronger connection.
Okay.
And that clustering actually triggers the recruitment of a whole bunch of other proteins.
Oh, wow. More players joining the game.
Yeah, you'll see names like talin, vinculin, paxilin.
Wait, those names ring a bell. Aren't those some of the proteins that link integrins to the cytoskeleton?
You got it. They form this like intricate network of interactions,
Right.
physically connecting those integrins to the actin filaments,
Okay.
which make up the cytoskeleton. It's like building a bridge, you know?
Yeah.
Spanning that cell membrane, linking the outside scaffolding of the extracellular matrix to the cell's internal framework.
Okay, so we've got this sturdy bridge
Yeah.
connecting the inside and outside of the cell. But if focal adhesions are so strong, how can cells actually move? Wouldn't they be stuck?
That's the cool part about focal adhesions. They're dynamic.
Oh, okay.
Remember those temporary campsites?
Yeah.
Well, these construction crews, they can assemble and disassemble those anchoring points super fast.
Oh, wow. That's efficient.
It allows the cell to adjust its grip on the extracellular matrix and move in a controlled way.
It's like they're adjusting the tension on the ropes.
Yeah.
Loosening, tightening their grip to move around.
Exactly. And this whole process, it's regulated by a complex interplay of signals,
Wow.
both from inside and outside the cell.
Okay.
It's a great example of how cells actually sense their surroundings and respond.
This makes me think back to cell migration.
Mm-hmm.
It seems like a really delicate balancing act.
It is.
The cell needs to be anchored securely to maintain its structure, but it also has to be able to release and move when needed.
Precisely. And integrins are, at the heart of that balancing act.
Yeah.
Modulating their grip.
In response to all kinds of signals.
I'm curious, what kinds of signals are we talking about?
Yeah.
What tells the integrins when to hang on tight and when to let go?
Well, it's a complex web of things, really.
Yeah.
But one of the key players is mechanical force.
Oh, interesting.
You see, when cells experience mechanical stress, like stretching or compression,
Yeah.
it can actually trigger changes in integrin conformation
Really?
and activity.
So it's like integrins are literally feeling the force.
Yeah,
And adjusting their grip.
exactly. And this ability to sense and respond to those mechanical forces, what we call mechanotransduction, is crucial for so many things.
Wow.
All sorts of processes in your body.
Give me an example. How does this mechanotransduction thing actually work in real life?
Okay, let's take bone remodeling.
All right.
When you exercise and you're putting stress on your bones, it actually triggers those integrins.
Yeah.
On the bone-building cells, the osteoblasts, to become activated.
Oh, wow. It's like they're listening to those signals
Right.
from their surroundings
Yeah.
and responding by, strengthening that bone.
So my workout's actually talking to my cells through these tiny integrins.
It is.
That's wild.
And then on the flip side, if there's not enough mechanical stress, the integrin activity goes down.
Oh, go.
And you might actually see less bone formation.
So that's why it's important to stay active for bone health.
Exactly.
Got it.
It all comes back to those integrins.
That's a great example of how these tiny interactions at the molecular level have real-world impacts on our health.
Yeah, definitely.
I have to ask,
Mhm.
how do scientists actually study these processes? It seems impossible to actually see these tiny integrins doing their thing.
It's definitely challenging, but researchers have come up with some ingenious techniques.
Okay.
One common method is a cell adhesion assay.
A cell adhesion assay. Okay.
They basically recreate that molecular handshake we talked about between integrins and the extracellular matrix in a controlled environment.
So like a miniature laboratory stage where they can observe those interactions.
You got it. They coat surfaces with specific integrin ligands, then introduce cells, and watch how well they adhere to that surface.
Oh, okay.
This lets them study the binding preferences of different integrins and test how different drugs or treatments might affect them.
Clever. It's like running a controlled experiment to see which integrins prefer which dance partners.
Exactly. Another powerful tool is immunofluorescence microscopy.
Okay.
This uses fluorescently tagged antibodies that specifically recognize integrins.
Interesting.
So researchers can actually see their location and distribution within those cells.
Oh, so it's like shining a spotlight on those tiny integrins.
Yeah, exactly. They can see where they're concentrated, how they're organized at those focal adhesions, and how their distribution changes in response to different things.
It's like taking a snapshot of the cell's inner workings, revealing that intricate dance of these molecules.
That's a great way to put it.
This is all so mind-boggling. It really makes you appreciate the complexity
Yeah.
of what's happening inside our bodies all the time.
Right.
And it sounds like there are some amazing tools for studying them.
Absolutely. And the more we learn about integrins, the more we realize how crucial they are for, a wide range of biological processes, from development to wound healing to immune response.
It's like we're peeling back the layers of this fascinating onion, each layer revealing even more complexity.
Right.
But I'm also thinking about what you mentioned earlier about malfunctioning integrins.
Yeah.
Being linked to diseases.
Right. And that's why this research is so important. By understanding how these things work, we can start to figure out what goes wrong and how we can develop new therapies to target those malfunctions.
So let's shift gears a bit and dive into that darker side of integrins. What are some examples of diseases linked to integrin dysfunction?
Well, one example is a group of bleeding disorders.
Okay.
Mutations in integrin genes can impair the ability of platelets to adhere to damaged blood vessels.
Oh, okay.
Leading to excessive bleeding.
So that molecular handshake is weakened or disrupted. And the platelets can't do their job.
Exactly. And then there are skin blistering diseases where those mutations disrupt the adhesion between skin cells, causing the layers of skin to separate, which forms those blisters.
Ouch, that sounds painful. It really shows how important integrins are for maintaining the integrity of our tissues.
Yeah, for sure.
But you also mentioned cancer before. How do integrins factor into that?
Well, in many types of cancer, tumor cells hijack those integrin signaling pathways to promote their own growth, survival, and spread.
So they're turning those cellular superheroes into villains. How do they do that?
Well, they can increase the expression of certain integrins that allow them to attach to and invade surrounding tissues. It's like giving them extra sticky fingers to grab onto healthy tissue.
And once they have a foothold, they can use those integrins to migrate to other parts of the body.
Right.
Forming those metastases. Scary stuff.
Yeah, it is. And that's why understanding how integrins are signaling in cancer is so important.
Right.
If we can figure out how they're manipulating these pathways, we might be able to develop new therapies to block them.
Got it.
And stop the cancer in its tracks.
That's a really exciting prospect.
Yeah.
It seems like integrins are at the forefront of so much important research.
They are.
But we've been talking a lot about the cellular level.
Yeah.
How does this research actually translate to real-world applications?
That's a great question. Integrin research is having a big impact in so many fields.
Okay.
Drug development, regenerative medicine, biomaterials engineering, you name it.
Tell me more. What are some examples of how this research is actually being applied?
Well, in drug development, researchers are designing new drugs that specifically target integrins to treat all kinds of diseases, including cancer, inflammatory diseases, autoimmune disorders.
So they're developing these targeted molecular wrenches to, tweak integrin activity.
Exactly. And in regenerative medicine, scientists are exploring how to use integrins to help cells grow and specialize.
So they're trying to harness those integrins to help repair and rebuild damaged organs. That's amazing.
It is. And then in biomaterials engineering, they're developing new materials that include these integrin binding motifs.
Right.
To improve how implants integrate into the body.
So they're designing materials that can talk to the cells, encouraging them to attach and grow on the implant surface?
Precisely. And that's just scratching the surface.
Wow.
As our understanding of these molecules keeps growing, we're going to see even more innovative applications.
This is so exciting. It really feels like we're on the verge of a new era in medicine, driven by our growing knowledge of this intricate world of integrins.
I agree. It's a fascinating field, and the possibilities really do seem endless. But with all this talk about manipulating integrins and developing new treatments, I think it's super important to consider the ethical implications.
That's a really important point. With any powerful technology, there's always a chance for misuse or unintended consequences.
Exactly. And it's crucial to have those open, transparent discussions about the ethical side of things, involving everyone, scientists, ethicists, policymakers, and the public.
Absolutely. We need to make sure this research is done responsibly and ethically,
Yeah,
with the goal of benefiting humanity and minimizing any potential harms.
I couldn't agree more. It's all about finding that balance
Mhm.
between scientific advancement and responsible innovation.
Well said. Now, before we wrap up, I want to circle back to something you mentioned before. How integrins can be affected by changes in the environment. We've talked about those mechanical forces, but what else can influence their activity?
You're right, those forces are a big one.
Yeah.
But it's not just about physical pressure.
Okay.
The environment around a cell, it's a complex mix of molecules, and some of them can interact directly with the integrins.
Oh, okay.
Like certain ions, like calcium.
Okay.
They're really important for integrin activation.
So the amount of calcium ions in the extracellular fluid
Mm,
can actually affect how well integrins do their job.
it can. And it's not just calcium. pH levels also matter.
Right.
Integrins function best within specific pH ranges. If it gets too acidic or too alkaline, it can mess things up.
That makes sense. It's like they're these finely tuned instruments, and even small changes can throw them off.
Exactly. And then there's the presence of specific molecules
Okay.
in the extracellular matrix. Remember RGD, that crucial handshake?
Yeah, yeah.
Well, there are other molecules that can either help or hinder integrin binding.
So it's like a big web of interactions
Yeah.
with all these different factors influencing how integrins behave. It sounds like understanding those interactions is key for developing those effective therapies.
Absolutely. If we want to change integrin activity for treatment purposes, we need to understand the whole picture.
Right.
It's not just about the integrins themselves. It's about understanding the environment and how those factors might influence their response.
That's a really good point. So it's not just about having the right integrins.
Right.
It's about having the right environment
Exactly.
for them to function properly. It's like setting the stage for them to do their job.
Precisely. And that kind of brings us back to that question from before. How do these environmental factors actually work at the molecular level?
That is a great question.
Yeah.
We've covered so much already, but there's always more to explore.
There is. As research continues, we're sure to discover even more intricate details about how they work
Yeah.
and how that all affects our health.
Well, I think we've given our listeners a lot to think about.
I agree.
We've explored the structure, the binding mechanisms, the role in various processes, their connection to health and disease.
Yeah.
It's been an amazing journey. And it's clear that this research is incredibly promising,
Yeah.
with the potential to change how we treat so many diseases. But, we got to proceed with caution. Think about those ethical implications of manipulating these processes.
Absolutely. We need that balance
Got it.
between the progress and the responsible innovation.
Exactly. And on that note, let's take a break.
Okay.
And we'll come back and explore some specific examples of how this is all playing out.
All right. Ready for some real-world stuff?
Yeah, let's hear it.
Integrin research is making huge strides in something we all care about,
Okay.
our hearts.
Oh, wow. You're saying integrins play a role in our cardiovascular health.
Absolutely.
Okay.
They're critical for building those blood vessels, keeping your heart muscle in check, even controlling how your blood clots. They're involved in pretty much every part of your circulatory system.
So they're like the architects and engineers of our cardiovascular system.
That's a great way to put it.
Wow.
Take angiogenesis, for instance.
Okay.
That's how new blood vessels are made, right?
Yeah.
Crucial for wound healing and tissue growth.
Right.
Integrins help those endothelial cells, the ones lining your vessels,
Yeah.
to move, multiply, and create those new paths for blood flow.
So if integrins are involved in building new blood vessels, what happens if they're not working right?
Well, if angiogenesis is messed up, tissues might not get enough blood.
Okay.
That's called ischemia.
Okay.
It can lead to some big problems,
Yeah.
like heart disease and peripheral artery disease.
Definitely want to avoid that. What are researchers doing about it?
They're looking at ways to, boost integrin function
Okay.
and get that angiogenesis going. Some are developing drugs that stimulate integrin signaling.
Right.
Others are looking into gene therapy
Oh, wow.
to deliver genes that code for integrins or their ligands.
So they're trying to give those integrins a little extra help to build those pathways.
Exactly.
What about the heart itself? You said integrins play a role there too.
Right. Integrins in your heart, muscle cells, the cardiomyocytes, they're crucial for keeping your heart strong.
Okay.
beating like it should, think of them as anchors, securing those cells to the extracellular matrix, giving them structural support and preventing that overstretching.
Wow. So they're literally holding our hearts together.
They are. They also help regulate the force of contraction, making sure your heart beats with the right strength and rhythm.
This reminds me of those focal adhesions we talked about. Are they involved here as well?
Absolutely. Those dynamic anchoring points are key for transmitting those mechanical forces and signals within the heart muscle.
So if those integrins in the heart aren't working properly, it could lead to heart problems.
Yeah, unfortunately. Okay.
Impaired integrin function in the heart can contribute to heart failure.
Right.
That's when the heart can't pump blood efficiently.
That's serious. Are there any treatments being developed that target integrins to, try to improve heart function?
Researchers are working on it.
Okay.
Some studies show certain drugs that target specific integrins might help improve heart function.
Oh, really?
They've seen it in animal models of heart failure. It's early days, but there's hope for new therapies down the line.
It's amazing that these tiny molecules can have such a big effect on our heart health. But what about blood clotting?
Yeah.
Remember, you mentioned integrins play a role there, too.
Right. We talked about how those integrins on platelets help them stick to damaged blood vessels.
Yeah, the molecular duct tape.
Exactly.
Okay.
But it goes beyond that.
Well, there's more.
Definitely. Integrins on platelets help activate signaling pathways.
That lead to the formation of a stable blood clot. They're also involved in, the balance between forming a clot and breaking it down.
Oh, wow. Keeping things in check.
Making sure it happens where and when it's needed.
So they're like the traffic cops of the clotting system.
Exactly.
Making sure everything flows smoothly.
If that balance is off, you can get bleeding disorders where clots don't form right.
Okay.
Or thrombotic disorders where clots form in the wrong places.
Right. Blocking things up.
Exactly. Both are pretty dangerous.
Yeah.
That's why understanding how integrins regulate this whole thing is so important for developing new treatments.
Wow. So integrins are truly multitaskers.
Yeah.
Playing crucial roles in angiogenesis, heart muscle function, and blood clotting.
It's this versatility, this ability to do so many different things.
Yeah.
That makes integrins so fascinating and important to study.
This deep dive has been incredible. It really makes you appreciate how complex and interconnected our bodies are, and how these tiny things can have such a huge impact on our health.
Absolutely. And as research goes on, we're only going to discover more. Who knows what other secrets are out there?
I'm sure there are many more discoveries waiting to be made. Well, to our listener, we hope this has sparked your curiosity about integrins. These tiny molecules are so much more than just sticky proteins.
Yeah.
They are essential in building our bodies and keeping our hearts healthy. And remember, this is only the beginning.
Right.
As scientists learn more about integrin biology, we can expect even more breakthroughs. Thanks for joining us on this fascinating journey.
Thanks for listening.